and this is a pre-lab lecture for the required AP Biology Lab dissolved oxygen. All right, so hopefully you'll have a good feel about what we're going to be doing in this lab so you can feel prepared and get the most out of it. So this lab is looking at something called primary productivity or primary production in an aquatic system. So we're going to simulate a pond. And you guys are going to be measuring dissolved oxygen. Dissolved oxygen is the oxygen that is available in the water for the aquatic organisms. So we know in a food chain that the producers are going to be our algae, or better known as phytoplankton. And then we have zooplankton, which are the primary consumers of the phytoplankton. So we have these trophic levels in aquatic systems just like we have in terrestrial systems. The difference between an aquatic system and a terrestrial system is oxygen. So one of the things that's really important is that you have to realize that it is a lot harder for aquatic organisms to get oxygen than it is for terrestrial organisms. There is so much less oxygen dissolved in water than there is available in air. So in surface water, there can be about 5 milliliters of oxygen per liter of water, and that's much less than the 250 milliliters of oxygen that we get in a liter of air that we breathe. And so aquatic organisms like fish have to have a system for concentrating that oxygen to meet their metabolic needs. And for fish, that would be the process of pulling in oxygen through their gills. And hopefully you'll have a chance to learn about this in class because there's some great physiology about how the circulatory system and the respiratory system of gills helps to bring in the oxygen from the dissolved oxygen in the water for the organisms. But oxygen demands, the minimum oxygen requirement is different depending on the organism. So some organisms like worms that live in the mud don't need very much oxygen. They can have a DO level of like 1 milligram per liter. But a fish like a striped bass would need 5 to 6 milliliters of oxygen, milligrams per oxygen per liter. So in fact, the desired level for aquatic systems is about 8 milligrams per liter. And we can also measure that in ppms, parts per million, or milligrams per liter, talking about how much oxygen is dissolved. And you see this is the desirable range. If you get less than 5 milligrams per liter, that's a system that doesn't have enough oxygen. And if it is less than about 2, that can be hypoxic, which means low, or it can actually be anoxic, which means no oxygen. And there are aquatic dead zones that happen in certain times of the year in the Chesapeake Bay system and also in the Gulf of Mexico, which we'll talk about. So how does oxygen get into the water? Well, there's a couple of different ways. One of the ways is just by mixing. So wind and waves and currents running over rocks, all of those things help to aerate the water and get oxygen in there. But of course, the other way to get oxygen into the water is photosynthesis, because as you know, photosynthesis is a chemical process where oxygen is produced as a byproduct. The problem is that oxygen that is produced by the producers is then turned around and used in respiration. So the aquatic producers use oxygen as respiration, and also the consumers use it. Furthermore, all of the decomposers that live in the bottom of the aquatic systems, the the fungi and the bacteria also respirate and use oxygen. So those are, you can see the demands of oxygen that we have in aquatic systems. So just to remember the photosynthesis equation, right? So we have three reactants. We have light, carbon dioxide, and water as our reactants. And then we have carbohydrates or glucose, and we have oxygen as a product. But remember that plants or primary producers do both. They do photosynthesis and they do respiration. And so they have their own little great system going. They can only do photosynthesis at night, during the day, because that's when the sun is. But they can respirate during the night or during the day, right? Because that's not dependent upon photosynthesis. So I want to take just a minute to talk about the difference between gross versus net. If you've ever had a real paycheck, you know there's a big difference, right? So your gross pay is the pay that you get before they take out all the taxes and the withholdings and all that kind of stuff. The net pay is what you have left over after they take out those deductions, all right? So we're going to see that we have a gross primary production and a net primary production. So the gross primary production, or GPP, is all of the glucose that is produced during photosynthesis. We say the carbon is fixed. So the carbon dioxide comes in and it is incorporated into some type of carbohydrate. That is our gross primary production. But just like your gross paycheck, we take some out, and what is actually left over for the other organisms in the energy pyramid is the net primary productivity. And so we have to take out the cellular respiration that the producer uses themselves. And so some of the glucose that is produced is taken out during cellular respiration, and the remaining glucose available that is going to be laid down as new material or biomass for all the organisms in the above trophic levels is called our net primary productivity. So you need to know these terms. Biochemical oxygen demand is how much oxygen these organisms need. GPP, I just explained, photosynthesis, respiration, and net primary production. It's very confusing sometimes because sometimes the text will say primary production and you don't know if they mean gross primary production or net, but they're usually talking about net. So we are going to use in our lab a method to determine dissolved oxygen called the Winkler method. And the Winkler method is a series of chemical reactions that ends up making a color change. And we are adding a chemical, a very specific amount in a process called titration. And in a titration, if you've had AP Chem, you know that when you add a known amount of a chemical that you can measure the volume very accurately, you usually put some kind of indicator in the chemical reaction that's going to turn color right at endpoint, right when the chemical reaction with this reactant that's in your titrant, that in your pipette that you have here, is going to equal in volume to the chemical that you're trying to determine that is in your sample. And that's what's going to happen with us in the Winkler method. So it's really fun. You're going to feel like a real scientist when you do this. We're going to add an indicator that's going to turn the starch in our sample blue. And then as we continue to add our chemical that's going to make a color change, when it turns clear, that's our endpoint, and that's how we're going to calculate our dissolved oxygen. So you're going to need to read your procedure, right? We're going to use these sample bottles to get some pond water. It's simulated pond water that we're going to get. And then we are going to add some magnesium sulfate, some alkyl iodine azide. We're going to mix it up. Then we add sulfuric acid, and we get some flock that occurs, and that's going to settle down. And then we put a titrated amount. We're actually going to use it in a little syringe. And we can see that it's very graduated here. And so we're going to get, tenths of a milliliter, which is going to be great. And we're going to keep adding. We're going to take our sample in here. We're going to pour a known amount into this container. And then we're going to titrate and swirl and titrate and swirl. And when this chemical turns the sample clear, that's our endpoint. And so then we're going to measure how much liquid we used, and that's going to get our dissolved oxygen. I'll explain that more in just a minute. So this lab actually has two parts, part A and part B. Part A, our experimental question is, what is the effective temperature on dissolved oxygen? So we're literally going to have three big buckets of water. One is going to be warm water, one is going to be cold water, and one is going to be room temperature, and we're just going to measure dissolved oxygen. Then we set up part B. Part B is actually measuring primary productivity, and we're going to change the intensity of the sunlight to see if we can affect how much dissolved oxygen is produced. Because our dissolved oxygen, because oxygen is also a product of photosynthesis, just like glucose is, we can tell how well we are photosynthesizing by measuring the amount of dissolved oxygen, right? Because that is another product in photosynthesis. So in part A, what is the effect of water temperature on dissolved oxygen? And then a corollary question is, what is the effect of water temperature on percent saturation? So we're going to talk more about saturation in the lab, but I want you to think of saturation as like a sponge, okay? When you clean up a big mess at the kitchen table and you got a lot of milk you spilled or whatever, at some point your sponge is going to be saturated. Can it hold any more liquid? No. So a higher saturation means that the water can't hold any more oxygen. It's not necessarily a good thing. So we'll talk more about that later. The way that you're going to calculate that is called, it's a system called a nomogram. And this red bar I have right here is just going to be any kind of straight edge ruler that you have. You are going to know the temperature of the water, and you are going to know the parts per million of your dissolved oxygen through our Winkler method. And so you're going to line up the temperature of the water with the dissolved oxygen that you determined. And then where it crosses this line right here, that's our percent saturation. All right, so that's how you're going to use a nomogram. In part B, we're going to actually, the first day we're actually going to set up our experiment, but then the next day we're going to do more titrations and determine some more dissolved oxygen of our samples. So our question here is, what is the effect of light intensity on the productivity of our pond sample? So this time we're going to have some bodies of water that have algae in them that I'm going to grow for you. It's going to be chlorella algae. And we're going to take some samples with our sample bottles of our pond water, which has algae in it. So they're going to be nice and green, and I will have put them under the lights to get them to be photosynthesizing. The way that we are going to change the light intensity is we're going to put a series of screens to make less light available through here. So if I've got algae in here, you can see if I put it under a light source, now I've got a little bit less light coming in. If I put 2 screens or 3 screens or 4 screens, I can gradually decrease the amount of light intensity, and that is going to be our independent variable that we're going to use. Is we're going to change that. I can also take this sample bottle and completely cover it with aluminum foil, right? Because then I can make it completely dark. So as you work through the Pearson Lab bench simulation, and I have a link later on the presentation, it's going to work you through part A and part B. You really, really need to do this so that you understand how to do the procedure and that you're ready to go for the lab when we do it. The main thing we need to know here is that the light bottle is going to have photosynthesis going on, but also respiration. So oxygen is being produced, but oxygen is also being consumed. In the dark bottle, only respiration is going on, not any photosynthesis at all, only respiration. So we have this great little equation that's going to help us. The GPP, the gross primary productivity, which you know is all of the productivity that the primary producer produces, including what it's going to use itself, minus what is respirated, the glucose that is respirated that was made by the producer in the gross primary productivity. What is left over is the NPP. And the NPP is very important because that is what is going to be the support for all the other trophic levels in our trophic pyramids to produce new biomass. All right, so NPP is equal to gross primary productivity minus respiration. So one of the things that we're going to do in our part B of our dissolved oxygen lab is that we are going to take the initial dissolved oxygen the very first day. So when you set up all your bottles with different numbers of screens, and some of you, of course, will have a control with no covering, and then some will have completely covered with aluminum foil. We need to, on day one, take the dissolved oxygen of our initial sample, because then we'll be able to compare that to how much dissolved oxygen, in this case, in the bottle that was exposed to pure light, or in this case, subtracted the dark bottle, which is only respiration, right? So this is how we're going to sneakily figure out our NPP, is we're going to figure out how much dissolved oxygen was used up in cellular respiration. And this is important, of course, because all this primary productivity in our aquatic producers, just like in a terrestrial system that we see here, this is where all of the energy, the chemical energy that is going to be used for all those other organisms is going to be produced. So I want you to think about how you can change dissolved oxygen. What conditions would make dissolved oxygen go up and what conditions would make it go down? And so what we're going to be looking for is our change in dissolved oxygen here. So remember, I told you that you're going to take a baseline sample of your pond water before we do anything to it over the night. And that's going to be important because this whole process, the light and the dark, that's going to be our GPP. And the bottle that's just in the dark is just going to be respiration. So we can get our change in DO, and that's going to help us determine. If you kind of look at some pond water from the side, if this was the surface of the pond, and then as I'm going down here in my water column, I'm getting deeper and deeper and deeper. I can see that in the photic zone, where light can penetrate, I'm going to have photosynthesis going on in here, right? And remember, photosynthesis and cellular respiration, right? So those producers are going to be doing both. But as I get deeper and deeper, I am going to be doing more respiration than I am photosynthesis. And way down here in the dark, I'm not doing anything but respiration, right? I'm not doing anything but cellular respiration. So there's going to be a point in the depth of my water column where my cellular respiration equals my rate of cellular respiration is equal to my rate of photosynthesis, and that's what we call the compensation point. Remember, all the nutrients are in the bottom of my body of water, right? Because that's where all the dead things fall down, and that's where the decomposers are recycling, and that's where my nutrients are. And we're going to talk about how we get the oxygen up here in the photic zone down to the bottom of where some of those organisms are, right? And how I get the nutrients up to where the producers are, because they need those nutrients to do photosynthesis. So the initial DO gives you a baseline for the experiment. If respiration is greater than the GPP, then you're going to have a negative DO. And you would expect that to happen more in the dark, right? And so I want to talk about this process of eutrophication. Eutrophication is a natural process, but as usual, the activity of humans can accelerate that. So we have two classifications of lakes. We have oligotrophic lakes and eutrophic lakes. In oligotrophic lakes, we have very clear water, we have high dissolved oxygen, we have low algae growth because things are going to be controlling that, and we have a lot of life in those. In a eutrophic lake, we have high nutrient levels, and that causes the algae to really kind of go crazy. And that causes less light to penetrate. Well, when less light penetrates, then I have less photosynthesis, and I'm going to have lower DO. And so what's going to happen is that some of these organisms, including the algae that don't have enough light to photosynthesize, are going to die, and they're going to filter down to the bottom, and the decomposers that live down here are going to start respirating, because that's what they do. They're going to be burning that fuel of the dead things, and they are using oxygen. So my dissolved oxygen is going to go down as my decomposer population goes up, because they're using the oxygen. So in a natural system, we talked about how we can flip this, right? So in the photic zone, I have oxygen being produced, and down here at the bottom, I have my nutrient-rich waters. And so there's a flipping that can happen with tides or winds or seasons that get that flipped, and that's what a healthy aquatic system needs. But as usual, humans end up messing this up. And we speed up this process of eutrophication, which is natural, by something called cultural eutrophication. And here's what happens. What happens is we can either get sediment that can run off, or we have organisms that, this could be just literally like dirt that can make the photic zone less clear and less likely to get sunlight. Or what usually happens is we have nutrient input. So we have fertilizers, nitrates and phosphates that run into this aquatic system, and it causes the algae to go crazy, right? Because they are utilizing these nutrients and they are photosynthesizing like gangbusters. But then their population gets so high in the photic zone that it cuts down the amount of sunlight. And what happens then is that they can photosynthesize less, they start to die, the other organisms are going to also be suffering because the decomposers are breaking down the dead algae and they're using up the oxygen. So there is an increase in the biological demand because of the decomposers, and there's going to be a decrease in the dissolved oxygen for everybody. And over time, that would accelerate this process of eutrophication. It can actually cause hypoxic areas of a body of water, or even anoxic, means no oxygen at all. So this is a good time while we're talking about trophic levels to just remind you that toxins can accumulate as we go up a trophic level. This is called bioaccumulation or biomagnification. So here's a chemical we'll talk about when we talk about our biochemistry unit, DDT. And we also have a problem with heavy metals like mercury that can also bioaccumulate as they go up the trophic levels. So I've got the address here for the Pearson Lab bench. My students should have that bookmarked. You're going to use it for every single AP biology lab. The chemistry of the Winkler method you do not need to know. It is in your lab if you want to look at it, but you do not need to know that for the College Board exam. What you do need to know is that when the material that we are titrating becomes colorless, that's our endpoint, and we can calculate then the amount of what we are adding in our syringe, and that is going to be equal to our dissolved oxygen. So just a little quick tip, when you get your water sample, you want to completely submerge your sample in the bottle in the sample of water so it's overflowing the top. So when you put the lid, liquid comes gushing out because you don't want any air in your sample bottle. And notice the fill line on the container here. You definitely want to fill your sample after you add your chemicals in the Winkler method. That's going to be your fill. And then you're going to take your sodium thiosulfate and you're going to pull up a volume of that. You must read the syringe correctly to know what your starting point is, right? And you can either read at the top of the plunger or the bottom of the plunger, whichever one you choose. Be consistent about that as you go forward, okay? So notice here, I have 0.35 milliliters here, all right? So we're reading very, very tiny amounts. And here's the money step. Write this down when you're in your notes. Every 0.1 milliliter of sodium thiosulfate, which is the chemical you're putting in your syringe, is equal to 1 ppm of DO or 1 milligram per liter of DO. All right, so that's important. So just very quickly, day 1 is part A. You're going to do 3 samples of cold, room temperature, and warm water. And then you are going to use your Nomogram to figure out the percent saturation of oxygen in those 3 samples. And then you're going to set up your baseline of your pond samples, and you'll be assigned different depths, different amounts of screening you're going to put on. Some people will be all sun, some people will be covered in aluminum foil. And you're going to get that ready for the next day. Then while you're gone that day, I will put the samples underneath the light, and they'll sit there for 24 hours, and then the next day you'll come in and you'll do your titrations and calculate how much dissolved oxygen is in the different depths of water. So remember those important terms. Remember, just the dark bottle is respiration. The baseline DO is the DO in our initial bottle before we let it sit overnight. And these other equations are going to be important for you to do calculations. So I hope this has been helpful for you, and I will see you in class. Bye.